Hello, greetings everyone. I hope you're doing well and are all welcome. In today's second class, Ms. Nurpur Akhtar, please go ahead. Floor for you.
Assalamu alaikum and a very good evening to everyone. Welcome to day 2 of our 3 days international training workshop on scientific writing excellence organized by VitaNova International Alliance for Sciences. Thank you all for joining us again and for your commitment to learning. Yesterday, we focused on the foundations of scientific research writing. Today, we move on a step deeper. Good research is not only about ideas, it's about data and, more importantly, how you analyze and present that data. Before we begin, a quick reminder. All sessions follow Pakistan time. Please adjust according to your local time zone. 100% live attendance is mandatory for the certification, and for attendance you have to write your serial number, and as you write your serial number for the secure attendance, kindly write your name with the serial number 3 times, first at the first of the live, then middle of the live, and the end of the live. Please maintain professionalism, mutual respect, and academic integrity throughout the workshop. Your active participation will make this workshop most meaningful. Now, it is my pleasure to introduce today's speaker, Mr. Muhammad Yasir Qureshi, founder of BioSkillz and affiliated with Bahauddin Zakariya University, Multan, Pakistan. He has a strong experience in data analysis and research skill development. Today's session is titled Advanced Data Analysis by R and Result Writing. This session will help you understand how to analyze data correctly and how to convert results into clear, publishable content. Without any further delay, I would like to invite Mr. Qureshi to begin today's session. Respected speaker, the session is yours. Kindly take the floor.
Hello, greetings everyone. Am I audible to all of you?
Yes, you are audible and clear. You can start your session now.
Okay, thank you, Nurpur Akhtar, for the kind introduction, and thank you, VitaNova, for inviting me today in this workshop. Today, what we will cover, you can say, what is the title of day 2, from raw data to meaningful results. I am Muhammad Yasir Qureshi, co-founder and CEO of BioSkillz, and you can symbolize, or if I am introducing myself in one line, so you can say as research data analysis tools mentor. So without further delay, we can move on. Next slide, please. Okay, what we learned today: basics of research data. We learned 5 things, or we can focus, or we are going to focus on 5 things. Basics of research data, statistical analysis for publication, tools for research data analysis, and writing results section, and fifth one is hands-on activity. Since to start with the basics, the first and main point is basics of research data. Research data is, what is data? Data is the collection of variables, different variables. And before going into data, we must have knowledge about statistics. What is statistics? You can remember, as you are not a statistician mostly, and I, as per my information, many students present here are from life sciences. So you are not statistician. You do not need to learn complete statistics to write your result section, to draw conclusion, to interpret your result. You just need somehow some things about statistics to fulfill your research. Keep remember four things about statistics. In statistics, we collect, analyze, interpret, and visualize our result. The first step is collect. We collect our data, and second step is we analyze. We analyze the pattern, insights about our research data, and third thing is we interpret by applying various statistical tests, and fourth one is we visualize our data in our research project, thesis, research paper, research article. Is that clear to all of you? Do tell me in chat box. Is that clear to all of you?
Dear participants, kindly write the answers if someone is asking so that we can know what you are getting or what you are not getting so that we can repeat that again. Thank you.
Thank you everyone for the response. And now just I share brief examples of research, or you can say research pattern, research flow. In research flow, firstly there is population, and the second is we can collect sample from our population. Third one is the treatment or the thing, or you can say the changes or association or effect we are going to do in our experiment. Fourth one is descriptive statistics, and fifth one, our main, which tell us helps to inference about our data is inferential statistics. Sixth one is observation. We observe pattern, relationship, comparison, correlation in our data, and then in the last we move to conclusion. We write our discussion section, and we write the results of our research article, research project. Next slide, please. Now, let's start about data. As I said, data is the collection of variables, different variables. What is data? Data is the collection of different variables at the time. So, variables are of many types, but we, as a life sciences student, we are going to deal with two type of variables: categorical variables and numeric variables. You all see Excel data. In our Excel, data is present in rows and columns. Am I right? In the Excel form, data is present in rows and columns. Am I right? What do you say about that, participant? Do let me know. In the Excel sheet, data is present in rows and columns. Yes, 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 thank you. So, but to analyze your data, you must remember rows are also called as observation, and columns are also called as variables. Okay, we can call rows as observation and columns as variable. Okay, now if I am saying variable, it means we can say it as column. Now the data present in our column is two types: categorical or numerical. What about the categorical data? Suppose my name is Yasir Qureshi. So my data type is categorical. The name is categorical. Okay, is that clear? But if our data contains 1, 2, 3 numbers, mathematical numbers, numeric number, so our data type is numerical. Categorical and numerical are further divided into two types. Numerical data is discrete and continuous also. What about discrete? Discrete means There is no decimal point in our data. Suppose 1, 2, 3, 4, 5. This data is discrete. It is numeric, but it is the type of numeric and it is called as discrete. Is that clear? Move on to continuous. The difference is simple. In continuous data type, there is a decimal point. Suppose 3.2, 3.24, 4.1, 4.3, 4.5. So the data is continuous when there are numbers present after the decimal point. Is that clear? Now, what about the categorical? Categorical means your data contains, simply the trick is your data contains ABCD alphabet, and the type is nominal and ordinal. Nominal means it contains no order words like man, woman, yes, no, true, false. Okay, someone name like Khalid, Tahira, Khalid, Fatma, Yasir. These names and the examples I listed before are included in nominal. What about ordinal? Ordinal, by their name, it means it includes order. For example, the data we are dealing in over survey data. Survey data is present in. Have you ever filled the survey form? Anyone? Have you ever filled survey? Survey include disagree, agree, neutral. Am I right? Yes, yes, yes. Agree, disagree, neutral, strongly agree, strongly disagree. Yes, I did, Fatma. You are right. So these type of data includes order like small, medium, large. Although it consists of alphabet A, B, C, D, and so on till Z, but this data is present in an order. That's why we called it as ordinal. Is that clear? Next slide, please. Now, in research, we can name our variables as independent, dependent, control, and co-founding. What are independent variables? Simply, the trick is independent variables are the variables that are in under our control, like variables which can manipulate by us. For example, if you are going to study the effect of fertilizer on rose plant, okay, and you collect 30 sample from your population. Now you divide these, you give 3 fertilizer to each of 10 groups. Is that clear? Now, what are independent variables here in our example? Independent variables is the fertilizer, is the type of plant. We select rose, but keep remember, rose is selected by us. Am I right? Means the researcher. Fertilizer ABC is selected by us, like researcher, me, you, and someone else, Noorpur Akhtar, Ali Hassan, Fatma. Okay, is that clear? So these are the independent variables. The variables, the categorical variables, the nominal ordinal variables that are selected by us. What are the dependent variables? Dependent variables are the outcome. Why we call them as dependent variables? Because these variables are dependent on independent variables. So actually, what is the workflow? Workflow is independent variables are selected by us and dependent variables are the outcome and they are dependent on independent variables. Is that clear? Just simple. You can remember them by their names. Independent variables are controlled by us, are selected by us, are manipulated by us, but dependent variables are controlled by, manipulated by, changed by the independent variable. Is that clear? Now, What are the constant variables? Suppose we are studying the effect of fertilizer on our rose plant. So in this example, what are the control or constant variables? Dear participant, do tell me what are the control or constant variable in our experiment. Do you have any idea? Do name the constant variables in our example. Muhammad Barham Khan said climate condition, control variable standard for comparison. Okay, you can say that, Fatma said. But in our example, what are the control variables? You should name them. Yes, you are right. Light, temperature. No, it's not kind of control group. Keep remember, it's not kind of control group. Although control group is the group in which there is no treatment. Okay, but controlled or constant variables are those which are equal in control and treated group. Okay, is that clear? Now, co-founding variables. What are the co-founding variables? These are termed as mediator variables. Okay, and these are the variables that are not included in our study but can affect our outcome. Okay, is that clear? Next slide, please. Now, simply to remember, you can simply take this example: cause effect, independent dependent variables. Keep remember, independent dependent variables. Independent variables is the cause and dependent variables is the effect. Independent variables is manipulated by us, by the researcher, me, you, okay, and dependent variables is the effect of independent variables and they are termed as measurement. Next slide, please. Now briefly, second term we are going to learn after the variables is population and sample. Simply, population is the collection of all subject, plant, individual variables or anything else on which we are going to study. Sample is the subset of our population. Okay, sample is the subset of our population. If we are collecting any sampling type is, you can and go with probability sampling, non probability sampling, clustered sampling, stratified sampling, random sampling. It totally depends upon the type of sampling, type of the research project, type of the research article or the conditions in which you are working or supervised by your supervisor. But the sample is the subset of our population. Keep remember, sample is the subset of our population, and the results, the conclusion we gain from our sample, the conclusion we gain from our sample is help us to conclude about our population, to inference about our population. Inference mean to understand, to make a conclusion about our population. Like if I am studying 30 samples from our rose plants, then what I will see that fertilizer A shows best growth among the all. Rose plant shows best growth among the all. So what we will say, if I applied fertilizer A to my whole rose population, I have best results. 95% sure that I have best results. Is that clear to all of you? Could you guys please stop typing your name and number in chat box? Just please stop. I guess it's for attendance, but it's one time. So please stop it and keep replying me. Nurpur Akhtar, if you are instructing them to stop this.
Uh, it is basically in the first of the meeting and in the middle of the meeting. So I do not know why they are doing it constantly. Kindly let me give them a reminder. Everyone, please remind that it is not the time to send your names and serial numbers. Kindly reply the questions. We will give you chance in the end of the meeting so that you can give your attendance. For now, kindly reply to our speaker.
Thanks, Nirpur Akhtar. Next slide, please. Now, the third thing we must remember before diving into our hands-on activity and other stuff, hypothesis. Hypothesis is the tentative statement. Okay, we are not sure about our statement, and hypothesis is the statement that we are going to prove. And if I am further specify the hypothesis, the alternative hypothesis. The alternative hypothesis is the basically the research question we are going to prove in our statistical test. Suppose there are many types of hypotheses, but the hypothesis we are going to face or we are going to deal with is null and alternative. Null means no difference, no association, no relationship, no correlation, no effect. Null hypothesis means the natural hypothesis, hypothesis with no effect. But on the other hand, alternative hypothesis is the main hypothesis, is the hypothesis which is proven by us or the hypothesis we are going to prove. Okay, for example, this as shown in the slide, the salary of men and women doesn't differ in Pakistan. But this is not our research question. Keep remember, null hypothesis is no difference hypothesis, no effect, no correlation, no difference hypothesis. But alternative hypothesis is the hypothesis that we are going to prove. And what is our alternative hypothesis? The salary of men and women differs in Pakistan. This is the hypothesis. I will give more example in upcoming slides, but for convenience, this is just a small example of hypothesis. And before going to test our results, we must need to highlight our alternative hypothesis. Keep remember, I am asking this thing later in the slides. Okay? Okay, members, participants. Thank you, Bharam. Kifayatullah, thank you. Mukarma, Nurul Fatma, thank you. Mehbiba, Mukarma, Taira, thank you. Now, the second, Hamza, thank you. Now, the second thing is statistical analysis for publication. We first done with the basics of research data, and we cover three things: variables, hypothesis, and population and sample terms. Now, the second part of today's session is basics statistics or statistical analysis for publication. You can name it as whatever you want. Before diving into, you must know about statistics as described by me earlier. So, could you guys please remind me what are four pillars of statistics I described previous in the chat box? Just please remind me what are the four pillars we are studied before in our first slide, or I can say it as you guys must need to remember these four things about statistics. Nothing need to go deep into these statistics. First one is collect. Yes, you are right. Second one is analyze. Yes, interpret and visualize. Yeah, that's great. That's great. Collect, analyze, interpret, and visualize. Yes, you are right. Same. There are many types of statistics, but We must need to know only two types of statistics, descriptive and inferential. Now, focus on the slide. Please, everyone, focus on this slide and keep remember what is the difference between descriptive and inferential statistics. Descriptive, by their name, is to summarize and describe main feature of our dataset. Okay, before diving into inferential statistics, that is the main statistics, that is the statistics of our concern, that is the statistics that helps us to inference about our population, the statistics that helps us to generalize about our population based on our sample data. We must need to know about the descriptive statistics. We apply descriptive statistics on our data, and descriptive statistics is further divided into three parts: the measure of central tendency, the measure of variability, and the skewness and kurtosis. Okay, measure of central tendency simply means mean, median, mode. Mean is the average of our data of our dependent variable. Okay, median is the center of our dependent variable when we are, if we arrange our data in an ascending or descending order. Okay, third one is mode. Mode is the most frequent term in our data. Okay, like the the data which is present again and again, the sample, the observation which is present many time in our data. Like if I am going through your comments in the chat box, so the person, there are many persons that are repeatedly answer me, so they are the mode, like they express themselves repeatedly, they are present repeatedly in the chat box. Okay, just for example. Now, the second one is measure of variability. The variability means the spreadness, the variance of our data, of our dependent variable, of our numeric variable, of our discrete variable, of our continuous variables. Measure of variability tells us about the spreadness of our data. Range, variance, standard deviation, IQR, quantile. We are not going into deep. Okay, we are not going deep about these terms, but I hope you guys have little bit knowledge about this. Am I right? Should you guys have knowledge about these terms, measure of central tendency and measure of variability? Come on guys, do let me know. Okay, that's truly great. I am happy, I am happy. Thanks for the for quick feedback. Now, move on, skewness and kurtosis. Skewness and kurtosis are the terms and relatively describe the same thing. Bilal Atas says that he has no knowledge about these terms. Bilal, due to shortage of time, I am unable to explain, but I hope you will get to know about these terms on Google and YouTube as well. Okay. The third one is skewness and kurtosis. What is skewness and kurtosis? Skewness tells us about the density of our data. Is our data is normally distributed or left skewed or right skewed, or is our data is dense at the center? Kurtosis tells us the variability of our data. Now, what is inferential statistics? As I described before, it helps us to inference, to make conclusion about our population. So what we are going to do in inferential statistics, this is the main statistics in which we perform tests on our data. The types are parametric and non-parametric test. The types and further parametric and non-parametric tests are divided into t-test, ANOVA, chi-square, and furthermore. Next slide, please. Now, the question is before performing test. Next slide, please. Before performing test, what we need to do? Before performing test, what we need to do? We have knowledge about sample size, about normality, about homogeneity, about hypothesis, about objective of research, selection of test, and the two terms p-value and confidence interval. We are looking into it about all these things, but firstly we are going into p value and confidence interval. Next slide please. What is p value and what is confidence interval, and how we set our hypothesis of our research data? Should when anyone have any experience of research in their MPhil or PhD? Anyone here have any experience, or he or she have to do his or her research data analysis on his own? Next slide please. That's great, Hafsa. Yes, that's great. Okay, Khalid, no issue. Okay, that's great. Yes, I okay, no issue. That's great, guys. If you have knowledge, but Ruhail Islamic BS thesis, okay, that's great. It's totally fine. Okay, but what I said to my students in my previous sessions, or you can say in my lectures, that you guys must need to know about these statistics mainly. That's enough. If you know the statistics terms and rules and regulation, then it's easy for you to apply statistical test, to select statistical test, to interpret your results, how to write your results. These are very small things if you guys have knowledge about these statistics, and if you are further refining how much statistics you should need to know is the biostatistics, how to apply statistical test, how to select statistical test, which type of test you need to follow in your data, how to arrange your data, how to find missing values, how to visualize your data. Okay, these are the basic things, you can say. If you are a researcher, if you call yourself as a researcher, if you are going to do research, okay, these are the basic things. Is that clear to all of you? Is that clear to all of you? Thanks for the response, guys. I hope you are enjoying the class. Now, now, it's the time to set up hypothesis, to set up hypothesis of your research. Okay, it's time to set up the hypothesis. Next slide please. Now, there are 5 steps to test a hypothesis. The first step is set up hypothesis. Second step is set up a significance level. Third step is test criteria. Fourth step is doing computation. Fifth step is making decision. Like it's a simple research flow or working flow of a statistical test analysis. Okay, first you set up hypothesis, second you set up significance level, and third you set up a test criteria for doing computation, fifth making decision. I am explaining these steps in our practical session after this theory. Now, how you can interpret your results? Simply you can interpret your results by most commonly rules by using statisticians and most researcher is p value. P-value. P-value, if P-value is less than your alpha, your significance level, you set up in your second step of hypothesis testing. If P-value is less than your alpha, you reject null hypothesis and you can set it as your results are significant. If your P-value is greater than alpha, we fail to reject null hypothesis. Uh, Below, here is a small matrix. You can say it as matrix or conclusion. Like if you accept your null hypothesis, what will happen? And there is a two type of error, type 1 error, or you can say as false positive, and type 2 errors, false negative. I hope you guys have knowledge. Those who done their research have knowledge about these two errors. Now next slide please.
Hello, Yasir. Is I'm audible?
Yeah, yeah, you are.
Okay. So class is continue. hmm Hello.
Yes, yes. Okay. Now, probability and confidence interval. What is meant by probability and confidence interval? I will explain it. What is meant by the main thing I explained that you should set up significance level in second step of your hypothesis testing. So probability and confidence interval. Probability denoted by P is the chance of occurrence of wrong decision, the chance of occurrence of wrong decision. And Q, what does Q means, or confidence interval means chance of right decision. Usually the sum of P plus Q is 1, or usually, or you can say most commonly we should need to follow this rule that P plus Q is equal to 1. Following in the table, I will explain some of the probability level the most researchers use in their research, and most commonly is 0.05, the second one. And in this case, the significance level is 5% and the confidence interval is 95%. So what does 95 confidence interval mean? Should anyone explain me in the chat box? What does 95 confidence interval means? I said that confidence interval is the chance of right decision. So if I said that 95 confidence interval, what does it mean? It means I am 95% confident that my results are okay. Am I right? Am I right? Do let me know, guys. Am I right? Like if I am saying that the my confidence interval is 95%, so what does it means? It means I am 95% confident about my results, about my conclusions. Okay, decision is right. Okay, less probability of error. Okay, yes, come on, do let me know. Now, 95% confident that my results are true, but what is the chances of wrong decision? The remaining 5%. Okay, is that clear? Yeah, Nimra said you told right that 5% only error are there. That's great. Next slide, please. Thank you so much, guys, for your response and for active participation. Now, this is the slide. I guess you guys must take screenshot of this slide. Must take screenshot for your future. If you are going to do research in upcoming time, year in a year, in a 6 month, in a month, or in your MS or PhD, this selection of test, how you can select your statistical test. This is the main thing. This is the thing we are, you can say, the purpose of today's class. How you can select statistical test. This is the conclusion, and just keep remember three things. First, you know about the before performing test. First, you know about the sample size of your data. Second, you know about the dependent and independent variables present in your data. Third, you know that either your dependent variables are homogeneous or normal, or they are not. Then, after knowing all these things, or descriptive stats also in the fourth step, the fifth step is the now you should move on to parametric test. If your data is normal and homogeneous, and if you have numeric data, dependent data, if you have categorical data, then you move on chi-square. Further, chi-square is divided into two types: one sample dependent chi-square test and two sample. And if your data is not normal, not homogeneous, then you should move to non-parametric test. Parametric and non-parametric are defined by their name. Parametric means the parameters. Parameters. Like what does it mean? Parametric tests are the tests that follows the parameters. What are the parameters? The parameters are the data is normal and data is homogeneous. Can you please, guys, can you please write this thing in chat box? What are the parameters of parametric test? What are the two parameters of your data, or your data must need to follow these two parameters for parametric test? Can you guys please write in the chat box what are the two parameters of your data, or your data need to follow these two parameters if you are going to apply parametric test on your data? Normal and homogeneous. Yes, you are right. Homogeneous and normal. Yes, yes, you are right. Now, next slide, please. Summary of parametric test. This is just a summary. Just take a screenshot of this slide. Very important to remember for you guys. When you are going to apply parametric test is clear if your data follows two conditions, normal and homogeneous. But which parametric test you need to apply on your data is defined by this chart. The chart shading on the screen, or you can say flow diagram. If you have two continuous variable, you select correlation. Pearson correlation in case of parametric test. In case of non-parametric test, you select Spearman. If you have one variable, you will go for one sample t-test. If you have two independent variables, you can go for two sample t independent t-test. Now, the third, if you have three or more variables, you can go for ANOVA, one factor, two factor, three factor, and Mankova, multiple factor. Next slide, please. Summary of non-parametric test. Same opposite to parametric test. These are the few changes that occurs in parametric test and will lead to non-parametric test. The conditions of non-parametric test are your data do not need to follow the assumptions, the rules, the conditions, and the conditions are normal, non-homogeneous. Non-parametric test types are one variable Wilcoxon and Mann-Whitney, two variables Mann-Whitney, and third, one or three or more variables. Then you have Crucier-Wallace-Friedman test also. Next slide, please. Tools for research data analysis. Now, there is a brief description of the tools of research data analysis, and I will explain. There are most commonly three tools that are being used worldwide for research data analysis or for research statistical analysis. What are these three rules? Microsoft Excel, SPSS, and RStudio. I will explain a few conditions, or you can say terms, why we use these tools, and what are why and what and how we use these tools. Okay, next slide, please. I am not going into detail because these things are written on slide as well. Rewriting results section is the main thing. Like software gives you numbers, but the real researcher turns these number into precision and describe it accurately. This is an art to describe your result. If you are master in using these tools, that's great, that's fine, but you do not know how to present, how to write, how to precisely write, or how to write in scientific way your results, you are not a real researcher. So should we start structure of the research section, or you guys feel bored? Tell me, should we start, or you are enjoying class? Thanks everyone. for active participation. Okay, start our studio. Mohammed Barham, just a few minutes away. We are going to start our studio. Just few minutes away. So keep patient. We are going to start our studio in a while. The structure of the research section. The structure is logical workflow is written and always report things. Do not include things are present in this slide. I hope you guys understand these terms, but for your confidence, I just quickly overgo from these things. In a logical workflow, you just firstly in a result paragraph, you firstly describe your dataset. Then you describe few characteristics of your dataset. The characteristics terms as descriptive statistics. Now, third one is your outcome. What is your outcome? Secondary, and if you are going to compare group wise, mention it. And the fifth part is trends or patterns. Always report effect size, confidence interval, p-value, direction, and reference. Just keep remember, results and discussions are two main things and two different things. Two main things and two different things. Results include what you interpret or what you get. Sorry, what you not interpret, what you will get from your results or from your statistical test. But discussion includes what and how you interpret and compare with previous results. Is that clear? The difference between results and conclusion. Do not include interpretation in your results. Do not include literature comparison or conclusion in your results. These three things are included in discussion part. Next slide, please. Tables and figures best practices. Always include title, axis, exact number, your output in visualized form. Okay, these are the simple things, but keep remember when to use table and when to use figure. If you have more than multiple, if your data is multiple variable, then must include table. But if your data is concise and bivariate and include three variables, two variables, four variables, then go for figures. Show your result in figures form. Okay, next slide please. Now, please take screenshot of this slide as well. Please take screenshot. I must recommend you to take screenshot of this slide as well. Like what writing and interpretations of your results clearly. I just said it. You can carry the logical flow to write your result section. And what is the flow of your result paragraph is first state comparison, second report numbers, third include CI, fourth is p-value, and fifth is direction. Now, for example, I will state it as the intervention group had a lower bacterial load than the control group, must include mean difference, confidence interval, lower limit, upper limit, and the p-value. Always use neutral language, be objective and neutral in your results, and if possible includes R ratio, odds ratio, mean difference, and R value. So next slide please. Is that clear? Guys, tell me, is that clear? We are, the wait is over and now we are moving on to hands-on activity. Confidence interval, Zikra Fatma, I will explain before. The confidence interval is the chances of right decision. Keep remember the chances of right decision. Okay. Now I'm going to share my screen. Just wait a minute. Okay, Nurpur Akhtar, should I share? I guess she is not here. Well, we will start. My screen is visible to all of you. Do let me know in the chat box.
Yes, your screen is visible. It's showing hands-on activity.
Yeah. Okay. Thank you. Thank you. L- Excel is the basic need. Moving on to R, I am showing just a brief introduction about the Excel as included in our topic as well. We do preliminary analysis. The analysis we need to do before doing our statistical test. Like we need to check out our data, we enter our data in Excel, we check descriptive statistics, we check variables. Like I select my whole data by pressing Ctrl A and I put borders on my data by applying Alt HBA. Keep remember these shortcuts Alt HBA, and I auto fit my column by Alt HOI and row Alt HOA. Okay, is that clear? Now my data is good, and to convert it into table form I select Ctrl T, and you can see here my data is has headers, and now our data is present in a beautiful way. Could you guys please tell me how many variables are present in my data? I will explain you in my class. Now please interpret and tell me how many variables are present in our data, in our current data. How many total variables are there? How many variables are there in our data? Could you guys please let me know about your understanding? Four, four. Okay. Four says I just let me know how many independent variables are there. There are four dependent variables and they are sepal length, sepal width, petal width. Is that visible to all of you? Sepal length, sepal width, petal width. Okay. Now, how many independent variables are there in our data? How many independent variables? Had Rohel said 8. No, Rohel. Four dependent variables are there. See, dependent variables are the variables that are in numeric form, that are measurements of our independent variable. So in this case, the numeric variables are these four. So what is the type of numeric? Is this discrete or continuous? Do let me know. Is these variables are discrete or continuous? These four independent variables are discrete or continuous? Yes, Subja Sarfraz, you are right. These are continuous because they contain points or values after decimal. Now, how many independent variables? There are one independent variables, and how many groups are there in our dependent variable? There are three groups: Zitosa, versicolor, and virginica. Okay, these are three plant species, and this is the practical data, or you can say exemplary data for our test. Okay, is that clear? For your convenience, I will explain how to find descriptive statistics in Excel. Just in a one click. You can go into data tab, then go into data analysis tab, and then go into descriptive stats option, click on OK, and now give the input range. I select my whole data by pressing Control Shift right, Control Shift down, and grouped are named by column. My variables are grouped by columns. Label in first row where I need to get output range. Output range here. I want my output here. Now summary statistics. Yes, nothing else. Click on OK, and in a few click you will get your summary of your data. This is the summary. See here, just in one click, you will get summary of your data. See here. Everything about your dependent variable is present here. Is that clear? Okay. Is that clear? You can get descriptive statistics in just one click from the Excel as well. Explain mean, median, standard error, kurtosis, skewness, range, minimum, maximum, sum, count. Okay. So this is the preliminary analysis, the basic analysis, the analysis we must need to do before diving into our main research. This is our studio. Okay, this is the R studio. We are going to do analysis on that. How to download this? Just go on browser. Right here R download. You can see this first website. Click on R download and click here download R 4.5.2 for Windows. Now R Studio as well. You need to download R Studio as well. R Studio desktop. Click on R Studio desktop, and you can see a website here. Now download R Studio desktop for Windows. Okay, is that clear? First, you need to download R and install it in your PC or laptop. Then you need to download R Studio. First R, then R Studio. R Studio is the best statistical software. Okay, the visualization, the statistical interpretation in R is recognized by many highest impact journal. Okay, your research paper is worth it when you include your statistical analysis or visualization from the R software. I will repeat again because someone said that, sir, kindly repeat. Just go on Google and write R download, and you can see first website here. Click on this link and your R will be downloaded. Download and install it first, then download. Click on R Studio download, write R Studio download, and then click on this, and you can see here option download R Studio desktop for Windows. Okay, is that clear? Now I close this one. I already installed R Studio as well. This is the basic interface. R Studio have three windows, or you can say it is closed as this. But if you click on this, you can see all the four panels. There are four panels. First one is the script, the place where we write code. We can also write our code in this console as well. Okay, but keep remember, the code we write in the console is not saved by us in our any file. But if we write our code in the script portion, we can save it for later use. Third is the environment. It can tell us about the connection, history, environment, the variables, the data present in our current script is shown here. Now these are the files, portion, plots we get here, packages, and everything else. So first of all, what you need to do, go here, or you can simply save this file by clicking on Ctrl S and go to desktop and save file as our international training, okay, by Sir Yasir. Okay, you can save this file as international training by Sir Yasir. Okay, now before going into, you must need to install packages. What is packages? Packages are the places that store functions. The function we need to perform in R are stored in packages, and these packages are present in repositories, online repositories. So to get those packages, we must need to install those packages in our session before writing code. So I will install few famous packages here. Install packages, and first one is tidyverse. I click here and click on run. So see here, this code is going to run until this red sign disappear. Now, this red sign disappear, and we install our package Now the second package we install is ggplot2. I click on run and the second package is going to install. Same, I will going to write. Third package is ggpubr, but when this red sign disappear, we are going to write this code. Now run this as well. Click on this run. Okay. We are also going to install packages read Excel and write Excel as well. Yes. Read Excel and write Excels are the two packages we need to perform function like how to import our Excel file and how to export Excel file. Okay, is that clear? Now packages are installed. How to active them? How to active packages in our current session? By clicking on library, writing library, and you can write all the packages name one by one here. The packages you need to active in your current section, just write here their name one by one. This is the basic syntax of R. Okay, now what is that mean? That this hashtag put make your written line in a comment. So if we run the code, these words started with the hashtags are considered as comments. Okay. Now the last one is ggpubr. I active these packages. Just wait a few minutes. You can active them one by one or by simply type library here. See, this is going to active, and if I am repeated again and again for every ggplot2 library ggpubr fourth one is library read Excel, fifth one is library write Excel. Is that clear till now? Everyone do let me know, is that clear till now? Should we move forward? We just install and active packages. Just put dashes before your comment, after your comment, and this is converted into a small chunk of code. Yes sir, clear. Okay, thank you. Now how to import Excel file? Import Excel file in R, because our data is present in the Excel file. So how to import this Excel file? Do not need to write any code, because we install the read Excel package. Just go here, click on import from Excel, and you can see this window. Now click on browse. My file is present in desktop. I click on book one and see the data present in our Excel is now going to import in our R file. So we need to set the data types of our columns. Sepal length, sepal width, and remaining are numeric. Okay, numeric. So I need to set them as numeric before importing. Yes, all set. Now I copy this code from here. Just copy this code from here. Click on cancel. Paste this code here as name your data as my data, and I click on run this command, and you can see our data is present. We can import our data. See here, our data is present. If you click if you click here, you can see your file. Or if you write view command and write my data, you can see your data. This is our data. Data can be stored in two forms, longitudinal and wider. Currently our data is present in wider form. Okay, now how to this is how to import excel file in R. Now how to export excel file in R. Is that clear? Is that clear to all of you? How to import excel file? Just click on here, import from excel, and you can go to browse your file position, and suppose, and just you can see this overview. Just set all the data types. Suppose it is numeric, set it to numeric, and now copy the code, paste this code here in the script, and you can see here how to export. Now this is the main question. Use command write excel xx and type your name of your data. Suppose my data is present, my data, and comma, I convert into iris data set dot xlxs. Okay, first name your data and then give the iris xlss format. I can click on OK, and this command runs. This file is saved here. You can see this file is saved here. Okay, is that clear to all of you? Now I close this one, close this one. Now we move to main part. I control shift I, and you can see here, this is the four things about data. Statistical tells us about the four things: correlation, distribution, comparison, composition. And what is descriptive stats? I explained already: measure of central tendency, measure of variability, and few things later. Descriptive stats. Now this is our data set. I name it as, you can say, our data is present here, and the file name of our data is my data. So we can import our data here. Moving on, this is our data iris, built-in data set already present in R. That's why if you write command data and then name of the data set, you will get your data as well. View command gives you the view of your data set. Iris numeric, I convert my data. I select on only numeric columns of my data to find correlation of my data. Okay, view iris numeric. Just close this. Now descriptive stats. How to find descriptive stats of your numeric data? This is the main task. I will explain you here. Descriptive stats, descriptive statistics. Now write mean. Just type here iris, and there are 5 variables. I will select only iris mean C. Here I will get, I click here, click on run, and I will get the mean of our data set. Is that clear? Is that clear? I will explain how to find mean. Is that clear? Now moving on next. Same is the case if you write median, iris, sepal length. Dollar sign gives you the specific variable of your data set. Same is the case. You can find mean, median, variance, standard deviation, IQR, quantile, quantile 1, 2, 3, minimum, maximum range. And to find the mode, you can install package modist as well. Okay, this is just a basic things how to find descriptive stats. Now I will close this inferential statistics. Keep remember this. I will explain also in my theory class, like we use t-test for three types and three purposes. Compare means of two groups: non versus over measurement, one sample t-test; x versus x, two sample t-test; x versus after time. This one is called as pair dependent. Okay, now if our sample size is less than 30, we use t-test. If our sample size is greater than 30, we use z-test. ANOVA, analysis of variance. Compare means of three or more than three groups: one way, two way, repeated measures, or we can say it as Mankova. After ANOVA, we use or we do pairwise comparison. We call them as post hoc test. Now, practically, practice of test. I already installed these. Now, this is the 4-5 steps we need to follow. We make our hypothesis, define our significance level, selection of test, conclusion, and decision. We move to first test, and first test is t-test one sample. We make our hypothesis. First step is we make our hypothesis. Okay, we make our hypothesis. Null hypothesis: there is no significant difference in both means. The alternative ones is there is a significant difference in both means. Okay, now define your significance level. Most commonly we use 95, 0.95. We set it as an 0.05. Selection of test: because we have only one dependent variable here, we select only sample length, so we use one sample t-test. And calculation: how to calculate? Simply just select your species or mark your data as species. I use dplyr here, IQR, or you can set it, I change it as my data. I change it into IQR1. Select yes, all done. I run this command, and further on I will apply t-test. So this is, we select only Sitosa species by using the select and filter. This is the statistical test we are going to get. One sample t-test. This is the basic syntax. If you guys are just mention your data set, and by $sign you can mention your data dependent variable. Second, then you can type mu. Because of the shortage of time, I am not going to type here the code. We can provide you the code. Okay, don't worry, we can provide you code. Because due to the shortage of time, I am not providing, or I'm not writing here code. This is already pre-written code by me, so I will provide you the code file by the organization VitaNova. Okay, don't worry at all, just keep the rules. Remember, I will explain here the syntax, the rules and regulation to write in our studio. t-test, t-test 1, this is our data set, and this is the sample length. Mu means mean over selected mean, 4.5, and alternative mean two-sided. Two-sided means they we can see here there is a difference or not. We are not going to see here either our mean is greater or less than. So this is our one sample t-test results. Now it says that true mean is not equal to 4.5. Our p-value is less than 0.05. So how we draw conclusion? I will explain you in decision part how we draw conclusion. See here, just follow these two rules. If our p-value is greater than alpha, we fail to reject null hypothesis. If our p-value is less than alpha, we reject null hypothesis. Okay, is that clear? Now according to rules, our p-value is less than alpha, 0.05. So there is a statistical significant difference between these two means. Now diving into two sample t-test independent. I separate my data, two sample. I separate my data for two sample, filter, view two sample. There is now I perform Shapiro test for normality. Yeah, Shapiro test. Our data is not normal. In case our data is not normal, we are going to do the non-parametric test. Here our data is normal. Our p-value is greater than 0.05. Now we perform t-test by using syntax already written here. Select our dependent variable, then select our independent variable, alternative, and data. You can see here this is paired t-test. We select pre-treatment, post-treatment, and to interpret how we interpret this is the main task. We can interpret it by the same rule. If our p-value is less than 0.05, we reject null hypothesis, and we said that there is a statistical significant difference between these two groups. Now the last one is ANOVA, and you can see here we check first normality, then homogeneity, and we can simply, this is the short command to apply ANOVA, AOV, select dependent variable, select independent variable, and data, and you can see here all this one. Is that clear to all of you? Is that clear to all of you? Do let me know. Is that clear to all of you? Now, should we make a little bit visualization here? Bar plot, box plot. Are you ready? Very tough to use. Brahm, due to shortage of time, I am explaining things quickly. Okay. Now, we are deep diving into visualization. Visualization here. I am just copy paste my code already written by me. Okay. This is, I will provide you the code. Don't worry. See here, we use ggplot, the famous, okay, the piping operator. Nimra said this is the piping operator. Okay, Control Shift M is the short key to write this piping operator. This pipe operator connects two or more functions. Okay, we can apply two functions here. First we call dataset, and then we apply select or filter function. Okay. Now this is the basic visualization we are going to do. I am just setting up my data. We need three main things: data, mapping, and the third one is geometry. I will set all here. You will get code. Now this is the visualization we will get. How beautiful it is. We can export it in 600 dpi, 1200 dpi. Okay, this is the visualization. I will provide you code, practice on it. Okay, now as explaining, moving on to our second plot, I will show you guys. Just I will set all things. Yeah, this is our second data set. It includes error bars. It includes bar plot of means of our groups. We are going to create this by using ggpabr. Okay, how we create is we only need three things: data, mapping, and geometry. We write it in code. I will provide you code. Just keep practice, and if you have an issue, you can contact me as well. You can contact me. Okay, if you face any issue, you can contact the organization. If you have any query, we will we will happy to answer and solve it. So before going, I will give you just one last one, you can say, visualization, the my favorite one. You I We will provide you code. Don't worry, guys. Don't worry about that. You can practice it on yourself. Now, see here how beautiful this plot is. I will provide you code. You can practice on it. This is the scatter plot. The three main plots we use in our research is bar plot, line plot, and box plot. Okay? Or this is the scatter plot. You can find correlation, linear regression, and other things also. But due to shortage of time, I am not going into that as well. We are just confined to this one. Okay, is that clear to all of you? I hope you guys enjoys the class. Don't forget to share your feedback. I hope you guys enjoys today's class. Don't forget to share your feedback. Yes, I am all done. I hope you enjoy. If you have any query, you can ask by the Muhammad Baram. You can contact me on my LinkedIn, Muhammad Yasir Qureshi, on my LinkedIn contact. Or if you have any query and any question, or if you guys need one-to-one personal classes on R programming, you can contact me as well on my LinkedIn, on my page, BioSkillz as well. Okay, or you can contact me through VitaNova by uh, Ghulam Murtaza and by Nurpur Akhtar. You guys can share my LinkedIn profile and details as well. Okay, I am all done. Thank you so much for active participation and for the, you can say, engaging session, great engaging session, and thanks to Ghulam Murtaza. Thanks to Ghulam Murtaza for inviting me, for giving me opportunity to speak at this platform international training workshop. And if you guys have any question, feel free to ask. Feel free to ask in the chat box, or if you are allowed to unmute yourself or anything, you can mute. I am available. Just go ahead.
Okay. I will take the floor now. It was great to have you on our live session, and thank you for your amazing, insightful, and informative slides as well as the hands on training. The unique thing was you made sure that audience and you have a two-way communication. That was a good point, and I really appreciate your effort by asking the audience time to time if they are listening, if they are getting your point or not. I hope that by that way they already got all the questions sorted out already. But if they have any questions, they can ask. But before that, I would like to invite um, Aksa Amin, the co-founder of VitaNova International Alliance for Sciences. Miss, if you are available, kindly take the floor. The floor is yours now.
I'm audible.
Yes, you are. You can
I'm
continue.
audible.
Yes, you are
Okay.
audible and
I
clear.
hope you're enjoying. Okay, okay, okay. I hope you are enjoying the classes of International Training Workshop, and I hope you learn about data analysis skills. Thank you to the Alliance Council unit members, Ms. Hafsa Sarfraz, Vice President, Mr. Mansoor, Deputy Director Social Media, Ms. Noorpur Akhtar, Head Collaboration, Ms. Neha, Technical Manager, to organize wonderful second class of International Research Writing Training Workshop with Mr. Yasir Qureshi, Founder, BioSkillz. Wish you great success in your future career to everyone. If you have any question regarding today's class, you can ask.
Thank you for the kind feedback, everyone in the chat box. As per direction, if you have any
Thank
query,
you
please.
so much, everyone. Thank you so much, Miss Aksa, and thank you so much, everyone, Alliance Council unit members. Thank you so much, Mr. Yasser, and thank you so much, everyone, core audience. I really appreciate your patience and your consistency for learning, uh, and I hope you learn. It is the basics of data analysis. I hope you will learn in deep context in future as well. So if anyone have any question, I hope you write down your questions. You can write in the chat box. Our moderator will announce your questions and we will answer your question. Thank you so much, Mr. Yasser, as well. I really appreciate your dedications, your time that we spent together. Thank you. Have a good day.
Uh, okay, I got a question which is, can AI write those code for us?
Mohammed Baran, AI can write these codes for us, but keep remember, AI is not supervised by us. Okay, he writes codes. AI writes code for us, but this code is correct or not, or directed as you ask, is confirmed by the expert. If you are expert in coding, then you can analyze, judge, and all the things that this code is correct and to use, and this code is meant for what you are looking for. Thank you.
Okay, I hope that you got your answer. The another question, which was earlier, that is, what is the most common mistakes researchers make when reporting results from R?
The main mistake is insufficient statistical reporting. Like most researchers consider that only significant results matter, but in science, non-significant results also matter. Okay, keep remember, as I told before, be honest in your research. What conclusions, what interpretation you get from your statistical test, you need to mention them as it is. Do not manipulate, change your results conclusions. Second thing is, just make difference between results and conclusion section. Do not include interpretation, do not include comparison in result section. Include what you will get. Only just include what you will get. How you interpret it, how you write it, how you compare it between it from previous results, you can write it in conclusion section. And for figures and tables, for figures, just be specific. Used highly defined images as R is most powerful tool here. Till now, its visualization is, you can say it as imported in 1200 dpi as well. Okay, so make your images clear, clear, and meaningful. I hope it clears all the queries.
Okay, if there's any question, anyone can ask in the chat box. I can read them. If you do not have any questions, we will suggest you to write your serial number and your name also for the secure attendance mark. And I would like to thank you, Mr. Qureshi, for answering the questions so clearly. I hope that no one has any questions because in the middle of the session you cleared everyone's doubts by asking them if they understood or not. I hope that that one made them to focus better and take the lessons. Dear participants, today's session was technical, so I strongly encourage you to practice what you learned today because there was a lot of hands-on practice also. Please remember tomorrow is day 3 focused on from manuscript to published papers. Session time 9 p.m. Pakistan time zone. Thank you all for your active participation. See you tomorrow. Assalamu alaikum and have a good night. Mr. Qureshi, thank you also for attending with us. We can end this live session now.
Thank you. And